Alright, welcome to this week's video. We are first going to start talking about the pressure volume curve for a single cardiac cycle. So I'm going to start and draw a little graph here. And this is pressure versus volume. That's what our graph looks like. And there's a line coming up here. So go through and label this. This is the spot where the AV node opens. This is when the AV node closes. This will be referred to as our inflow to diastolic. And atrial systole. Okay, and this whole space from the time the AV opens to the time it closes is referred to as stroke volume. And then we have this line will represent the end systolic volume and the end diastolic volume. So this part where the AV closes, we call this preload. So preload is all about the stretch or the volume of the blood. Afterload is right here. And afterload actually refers to the pressure in the aorta. Or another way you can think about it is the resistance in relieving. This period in the middle where there is no change, AV closed, this is the isovolumic. That means no valves are open. Okay, so this green line I drew over here, this is referred to inotropy. And what that is it's how hard the heart is working. Or something else we can refer it to is elasticity. And the formula for this is the change in pressure over the change in volume. The opposite to this is compliance. Compliance is change in volume over the change in pressure. And how this is regulated, we refer to it as something called MAP. MAP is the mean arterial pressure. Mean arterial pressure. And the formula for that is cardiac output times the peripheral resistance. So as we've learned previous, cardiac output is stroke volume times heart rate. And then peripheral resistance, there's a formula and kind of a good simplification of this is length times radius, or length over radius to the fourth power, meaning that peripheral resistance is highly dependent on the radius of the vesicle. So MAP is regulated by this, by stroke volume, heart rate, and peripheral resistance, which is the radius of the vesicle. So we have a way that the heart regulates this, and this is through baroreceptors. Baroreceptors are found in the aortic arch, and they sense this stretch of the aorta. And as they sense blood pressure rising, they make the heart rate go down and the peripheral resistance go down by sending a message through the parasympathetic nervous system through the vagus nerve to the brainstem. And in the same sense, it senses, so if it senses blood pressure is low, what it will do is it will increase the heart rate and increase the peripheral resistance through the sympathetic nervous system. By going through this, goes through the glossopharyngeal nerve. That's cranial nerve number 9. And this is more of a shorter term way to do that. So ways that we can affect our heart rate are, we have short term, these baroreceptors, as we've talked about, and then we have chemoreceptors. And these respond to things such as carbon dioxide, lactic acid, and oxygen. But a more long-term way that it uses this is when it senses mean arterial pressure is low, it releases renin. And as renin's released, it activates something called angiotensinogen, which activates Ang1, which then activates, I believe it's ACE, then activates Ang2, and Ang2 activates aldosterone, it makes you feel thirst, and also have vasoconstriction. Which, if we look at this from a holistic perspective, so thirst will increase our blood volume. Vasoconstriction will increase our blood pressure, and aldosterone as well helps with water reabsorption, so increases the blood volume. And so what this is important for is it's important to understand because ACE inhibitors are among the most common ways to regulate blood pressure. So most blood pressure regulating medications are ACE inhibitors. Now to talk about the last thing that we have to talk about today, we're going to talk about capillaries. So we have this. This is called, on this side, we refer to it as blood pressure. And on this side, we refer to it as hydrostatic pressure. And inside, on the edge of this, we have this barrier. This is called the precapillary sphincter. So we have a few sphincters in our body. They're all smooth muscle, which they close down and they regulate what comes in and goes out of our body. This capillary consists of these cells. They're tightly woven together that make up the walls. Then on our capillary, we have our blood cells, and we also have these proteins and plasma. 80% of what's in our body is albumin. And so we have a few values that we refer to as we look at the difference between the venous side and the arterial side. And we'll look at it and see how it changes. So a word that is important to realize and to understand is colloid. I guess it's not super important to understand, but I'll just give you a little bit of background. Colloid refers to floating particles. So this is a term that comes from chemistry. They use it for particles that float in water and they don't necessarily sink to the bottom. So we refer to this, we talk about these proteins, we call this BCOP. It is blood colloid osmotic pressure, and then we have interstitial colloid osmotic pressure, and within our capillary, this hydrostatic pressure is always wanting to push out. This BCOP is wanting to stay in, ICOP is wanting to push out, and then we have one more thing over here. These are lymphatic vessels. So what lymph nodes do is they take blood and particles, and then they recycle it right through the lymphatic system, and then they eventually cycle it back into the blood and into the bowels. There's a few things it does. We'll refer to this as interstitial fluid pressure. And so we're going to look at these two sides. So we're going to look at arterial versus our venous sides. So we have values. What we like to do is to refer to them. We talked about in and out. These lymphatic vessels are pulling it out. So we'll start there. Out. And so this is fluid pressure. And this is very low. This is only 3 millimeters of mercury. We have ICOP. Now first we'll go hydrostatic pressure. And we'll total it all up here. Okay, so on the arterial side, this hydrostatic pressure is 30 millimeters. And on the venous side, it's only 10 millimeters of mercury. For the ICOP, it stays consistent, and it is at 8 millimeters of mercury. And for BCOP, it stays consistent at 28 millimeters of mercury. And as we total this up, we can see that this will cancel, and our net will be 13. Let's see in the other color. 13 millimeters of mercury out of the cell. Having it to have that higher pressure. And as we look at this one, we can see that it's going to be more on the inner side as 28 overcomes, and it's only going to be 7 millimeters of mercury inside the cell, inside the vein. So we can kind of see this all working. What changes here is we mentioned we have these cells that line the blood vessel, the capillary. And something we can have is we have these mast cells. These mast cells have all of these things within them. And these things, these red circles, are histidine. Okay, and what histamines do is they open this up. They create a hole right here. And what that does is the BCOP, therefore, runs out and is lower. So if we look at this, if we have the BCOP lower on this, on either one of these, we will have a higher percentage out and a lower percentage in. So what that does, as we refer back to osmosis, water flows towards salts. And so if we have a higher percentage of these proteins and this albumin outside the cell. That's going to lead to edema because the water is going to follow it, causing us to have swelling. And this is often a reaction to allergies. And so that is about all we have to talk about today.